Okay, I'm just going to jump right in. And we know that phenotype is influenced by both genetic and environmental components. And in the context of EPDs, we're particularly interested in the proportion that's due to additive genetic variation. So from a genomics context, we can think about that as mutations within the genome which cause differences in phenotype. So for example, an animal might have extra growth as compared to another animal. And the amount of additive genetic variation is reflected in the heritability estimate of the trait. So for example, traits with low heritability like milk production and heifer pregnancy, which would have a much larger influence from the environment as opposed to additive genetics. Whereas if we have highly heritable traits, like a lot of our carcass traits, it's much more highly influenced by additive genetic variation than from environmental components. And again, for national cattle evaluation, we're interested in this black portion for traits which are influenced by large numbers of mutations or quantitative traits. And these are described by EPDs and are stably inherited from one generation to another. So if we consider additive genetic variation from a genomics context, let's say we have a mutation in a gene which we know affects ribeye area. So depending on the animal's genotype and the allele frequency and the effect of that mutation, these would be the contributions to the animal's genotype. So take-home message, if the animal is BB, she never passes on a favorable A allele. If she's AB, she passes it on 50% of the time. And if she's AA, she always passes on that favorable allele. And what I really want you to take home from this slide is that the combination of all of these alleles and their contributions towards the breeding value, or all the mutations within the genome, results in the genetic makeup of the entire animal. And if we knew every causal mutation, we can add them up, and we would know the EPD of the animal with an accuracy of 1. Unfortunately, it's not that simple because we don't know what these causal mutations are for the large majority, almost all of them within the genome. So again, we come back to this training concept. So we have a population of animals where we estimate the marker effects. And then we apply that knowledge in some sort of implementation population, which in this instance are going to be our selection candidates in the first generation after the training has occurred. And in this population, we predict the breeding value, and we know the accuracy of those breeding value predictions from the validation. Now, there's 2 ways that we can model these effects, and Allison touched on them briefly. I'm going to talk about them in a little bit more detail. The first is linkage disequilibrium, and this is found population-wide, and it's stable over many generations in a population. We also have linkage information, which are a function of animals that are close relatives. So they're describing familial relationships within the population. And this, we know, breaks down faster over generations than does linkage disequilibrium. And I hope in the next couple slides, it'll be clear why that is. So if we go to linkage and we look at the animals in the founder generation here, we pull 1 chromosome from each of the parents, and then we end up with 2 different colors here. So then we have another generation, and we break up those chromosomes, those colored blocks, the large blocks, we break them up even further by recombination within this generation. When we mate this generation together, we end up breaking up those blocks even more. So you can see how we start with large blocks within the genome, and over many different generations, we break those blocks up, and we end up with lots of colors down here, because we've broken up those blocks of linkage information. So you can see how that breaks down pretty rapidly over time. And that is in contrast to something like linkage disequilibrium, which we typically would find between markers that are close together within the genome. So they break apart more slowly over time, and we can use them across many different families. So as you might expect, these are the kind of effects that we would like to model. And as a function of how these blocks work within the genome, they have to have similar allele frequencies. So in order to get an idea, we have a marker here denoted by M, and we have a big M and a little m, so 2 different alleles. And we have a QTL, or a causal mutation, which is either a big Q or a little q for the 2 different alleles there. So if we always find big M with big Q, then they're in complete linkage disequilibrium. And we don't really need to know what Q is, Because if we know what big M is, we know that it's paired with a big Q. And the same if we have little m, we know it's paired with a little q, and we can predict that very reliably across the entire population. And this comes into play because from SNP genotypes, we can sometimes tell the exact genotype of the animal at the QTL locus. So for example, these animals would be AA, these animals would be AB, and these animals would be BB. And then you see these animals here in between, and those are animals where there has been a recombination between the marker locus and the QTL. And the same situation here, only we have considerably more recombination. So these markers are likely further apart from the QTL than these over here. Now in reality, what we actually have when we talk about training and validation is we have a training population up here. We can still do the validation and implement those genomic tests within our selection candidates in the first generation. Now ideally, we can take genotypes and phenotypes collected from these animals and recycle them back up into the training population, retrain those models, and then continue this process in kind of a circular fashion. Now, if we can't do that, what happens is you continue to use those same prediction models that we generated here, only we use them, maybe 3 or 4 generations later. So generation in, we apply those same marker predictions, but at this point in time, we really don't know what the accuracy is because we know that it decays over those generations. So the big question is, how large is the decline in accuracy? So we know that the predictions use linkage and linkage disequilibrium, and we know that they decay over time due to breakdown in those familial relationships, like those colored blocks I showed you, and also changes in allele frequency. And we know we can measure the decay, but it does require several generations of pedigreed populations or some type of simulated data where we know exactly the genotypes or the marker information as well as the pedigree structure. Now, in the absence of this data, we wanted to do a little experiment, and we took animals from the National Cattlemen's Beef Association Carcass Merit Project, and we used those animals to train our prediction models. So we had 50K SNP genotypes on 3,240 animals, and we used Warner-Bratzler shear force in this particular experiment. And for those of you that aren't familiar, that's an objective measurement of steak tenderness. And we had 5 breeds of animals within those 3,240 animals. We had roughly 600 Angus and almost 700 Charolais, a little over 1,000 Hereford, about 300 Limousines, and about 500 Simmental, to give you an idea of the breakdown of that population. And we just took the entire population of 3,240 animals, and we just randomly separated them into training and validation sets. So we didn't pay any attention to what breed they were. We just basically tossed all 3,240 IDs in a hat, and we pulled out, say, 70% of them, or 60%, or 90%, as it were. And we also had a second population of animals from the University of Illinois, which also had Warner-Bratzler shear force records. And there were 443 of those animals, so considerably fewer than we had in the carcass merit population. And these were comprised of Simmental and Sim Angus animals. They were about 8 to 10 generations more contemporary than the animals in the carcass merit project. So about 2 generations in time. One of the first things I did was build a genomic relationship matrix to look at the relationships between these 2 populations, because we know that a lot of the predictions are due to familial relationships. So this reflects the degree of relationship between those 2 populations. So in this block here, we have the original NCBA Carcass Merit Project animals, and you can see they're pretty highly related to one another. In this block down here, we have the animals from that external validation population, and you can see they're even more highly related to one another. When you look across the 2 different populations, you can see there's basically no relationship at all according to the genomic relationship matrix. And this actually looks like we built a matrix between 2 animals from 2 different breeds, like between Angus and Hereford, as opposed to animals from the same breed or similar breed makeups just stratified over time. One thing I do want to point out before I show you the results for this is that we don't know the exact number of generations that separate these two populations. We just know that there's about 10 years between these phenotypic records. And I say that because if these animals down here are direct descendants of the Carcass Merit Project animals, it's going to be about 2 generations. But if they're not direct descendants, we don't know the actual number of generations separating these populations to a common ancestor. So this slide's very busy, so I'm going to start kind of on the left and work my way to the right. So we've got a realized accuracy on the y-axis, and along the x-axis here, we start with 95% of those 3,240 animals in training, anywhere down to 10% of those animals in training, or where we have 3,240, we used all of those Carcass Merit Project animals in the training. Now these purple bars here are where we used whichever animals were not used in the training, but were part of the NCBA Carcass Merit Project in the validation population to test the predictive ability of the models. So that's reflected in the accuracy here. And you can see that we did, when we used animals to validate that were within the same original population, but were not part of the training set, they were mutually exclusive, we did a pretty good job generating these prediction models. They have an accuracy of about 0.6, using anywhere from 60 to 70% of the data on up. But if you look down here, these shorter bars are where we use the same animals in training that we used for the purple bars, but instead we did the validation within that external validation set, those other Simmental and Simangus animals. And you can see that we have about a 90% reduction in prediction accuracy. And it really didn't matter which type of model we used, other than that the base C-Pi model tended to perform better, but that's probably because it emphasizes the effect of calpain and calstatin, which are known genes with large effect on Werner-Bratzler shear force. So you can see over about 10 years in time, we had a really drastic decay in the prediction accuracy. Now again, I do want to emphasize that we have an unknown number of generations between these two populations. So one of the things we're going to go back and do is try and get the pedigrees for the sires of these animals. We don't have any pedigrees on the dams, but we can go back with the sires and figure out how many generations until there are some common ancestors between the sires from these two different populations. So from this, we were able to conclude that retraining, even after relatively short periods of time, is probably going to be essential for maintaining the predictive power of these models. So maybe we want to consider doing this every year, or if we could, maybe after every calf crop. I do want to point out that these models were built across 5 different breeds of animals, so the decay was likely worse than what you would find in a purebred population. But this is still going to be a major issue for national cattle evaluation, especially as it concerns evaluations of hybrid animals within the industry. Identification of causal mutations by sequencing or modeling using sequencing data should mitigate these effects, but as anybody knows who's tried to find causal mutations for quantitative traits, this is not a proposal to be taken lightly, and it's very difficult to do. And later on, we'll hear from Dr. Catchman on whether the 700K will be able to increase our prediction power. So I'm looking forward to that. So retraining periodically and possibly the related data ownership issues, whether we own the actual genotypes to do the retraining or not, is likely to be critical to make progress as we move forward in the future. And I think I stopped pretty early because I thought I had a 15-minute talk and I have a 20-minute talk, so I have time for questions.